This is an overview of eukaryotic gene regulation. Remember that prokaryotes respond to environmental signals, turning genes on and off as necessary, as in the case of the lac operon that we saw earlier. Eukaryotes, on the other hand, follow a developmental program, that is, they go from a fertilized egg to a multi-celled organism with many differentiated cells, and they do so in response to internal signals. Sometimes there are hormones, sometimes there are substances produced by neighboring cells during embryogenesis which dictate that a blastula becomes a gastrula and that a gastrula becomes a primitive embryo and then the embryo develops and so on. You may have seen a little bit of this in 150, this developmental programming. And to do this, genes get turned on and off, and we know that different cell types in eukaryotes, in multicelled eukaryotes, are in fact different because they are expressing different sets of genes. They may be overlapping sets of genes, but nonetheless, a muscle cell produces lots of actin and lots of myosin from actin and myosin genes, and a host of other proteins related to muscle activity, muscular contraction, that red blood cells and white blood cells don't produce, and red and white blood cells are producing other proteins necessary for their survival. We're beginning to think that a human being is actually the product of a blueprint of maybe 25 or perhaps 30,000 different genes. Now, that's not that many for organisms as complex as we are. And even with those 25,000 genes, no one cell expresses all of them at the same time. So different cells exhibit differential gene expression. And then, of course, cells also respond to internal and external environmental signals, nutrient levels, you name it, and the expression of genes is controlled by the chemical environment of cells. Ultimately, all of these signals will lead to changes in transcription. There are some examples of changes in translation rate even. As you'll see in a moment, eukaryotes go through quite a number of steps in order to go from the information in a gene to the information in a polypeptide. And control can occur at any one of those levels. And ultimately, gene regulation, or for that matter translation regulation, which is a kind of not quite gene regulation, can result in changes in protein levels and therefore changes in a cell's metabolism. Most of what we will look at is transcription regulation because that's largely how we understand gene regulation to occur. And it happens in the following general manner. As with bacteria, there are regulatory proteins produced by regulatory genes. Now, eukaryotes don't organize their genes into operons, so if there is any coordination to be accomplished that is regulating comparable genes in the same pathway in the same way, it isn't by the organization into operons. Rather, genes that must be regulated in some coordinated fashion will share DNA binding sequences, that is, DNA sequences that can bind to transcription factors. There are transcription factor binding regions in DNA, and then transcription factors that bind to them that results in the regulation of those genes. We recognize spatially two kinds of DNA sequences that recognize these transcription factors. There are promoter proximal ones, meaning DNA sequences not too far from the promoter, the region where RNA polymerases are going to attach to begin transcription. And then there are those that are distal, meaning some distance away from the promoter. And as you'll see, that distance can be quite considerable. And we'll see why that might be of some use in regulating eukaryotic genes. Transcription factors are more numerous and more varied in eukaryotes than they are in bacteria. Here's an experiment. Remember a moment ago I said that we have 25, perhaps 30,000 genes in a human and about, say comparable numbers of genes in other higher organisms, other mammals and so on. And the different cell types are different because of the different sets of genes that they are expressing at any one moment. Let's see if that is in fact a reasonable way to explain cell differentiation. You can take an egg and remove its nucleus. Let's do that. Originally it was done for a toad, a xenopus, a relative of a frog, and more recently, of course, it's been done with higher organisms. So now we have what's called an enucleated egg, an egg without a nucleus, and we can take the nucleus of an adult cell and transplant it into the egg. And here's what happens. The egg begins to develop, and under the right conditions, and with a bit greater complexity than I'm showing here, this egg with an adult nucleus in it instead of its normal nucleus can develop into a normal adult. And this is the essence of cloning in frogs and sheep and cows and more recently cats and dogs and you name it, and eventually I'm afraid also humans regardless of the ethics of the whole thing. But this is the essence of cloning organisms the way you've read about this in newspapers or seen it in the specials on TV. So you can conclude from this kind of experiment that the nucleus of all cells of an organism, in the case of the frog it was an intestinal cell, but any cell can be used pretty much, all cells, all somatic cells of an organism contain the same genes as you would find in a fertilized egg. I mentioned earlier that there is a complex series of events that a eukaryotic cell must go through to produce a messenger RNA. And at every step of the way, you can have some aspect of gene regulation. Chromatin remodeling, going from heterochromatin to euchromatin, involves a number of discrete chemical steps, and if you could control those steps, for example, control the acetylation or phosphorylation of histones, you could control whether genes are active or inactive in the vicinity of those histones. And that does occur. During development, previously heterochromatic DNA packed around silent genes might need to be opened up in order that genes that had not been expressed to that point are available for expression so that cells can differentiate. Then, of course, we have transcription, and we make a pre-messenger RNA, and then you saw that we have different kinds of transcript processing. If it's a ribosomal RNA, it undergoes a cleavage-based processing. And here we have pictured mRNA processing, which of course involves splicing, 5' end capping, and 3' end addition of a poly-A tail. Those are separate discrete steps, any one of which, in theory at least, is subject to controls. Molecules in the cell or in the nucleus that speed up addition of a tail, slow down addition of a tail, increase the rate of capping, and so on. And capping, remember, is one of these processes that are thought to enable the exit of a mature message out of the nucleus and into the cytoplasm where it will be used. So if you could control that, you would control the amount of final protein product that this gene could make. You may remember the poly-A tail has something to do with the number of times that a message is actually transcribed by ribosomes. So if you could make shorter tails at a given time for a messenger RNA, you will end up making fewer polypeptides during the translation process. And these are all just nuclear events, and then of course the message has to exit the nuclear pores and get into the cytoplasm. That's a step in itself, which in theory could be regulated. And then once in the cytoplasm, ribosomes have to bind, and we know that, as I pointed out earlier, that translation can in fact be regulated. There are a few examples, which I may or may not have a chance to go through at some point. But just be aware that that's a point at which some controls over the final amount of protein produced can be exerted. There's translation. There's also mRNA degradation. Well, nothing lives forever. No molecules live forever. RNAs that are produced in eukaryotes have varying half-lives, meaning varying lifetimes in the cell. There are some that are very long-lived, and others that are degraded within minutes of being produced and released into the cytoplasm. All evolved, of course, to make sure that the right amounts of proteins are produced, and no more and no less. Prokaryotes typically make short-lived messenger RNAs. So all of the proteins they need to make, in order to keep making them, the bacterium has to keep transcribing the genes. Only those genes that are regulated can be shut off so that their proteins are not made unless they're necessary. Eukaryotic cells, being specialized, have the luxury of shutting many genes off, in effect permanently, and then regulating those genes which it must express in an exquisitely intricate fashion so that it produces the right amounts of proteins for the right amounts of time, both for normal life and in response to environmental signals, meaning signals from the blood, the fluid around the cells, or even from external signals, and then of course timed signals, developmentally programmed signals. There we have other aspects: protein folding, transport, and activation. Remember that proteins have to assume their correct three-dimensional shape, and this is often facilitated by proteins called chaperone proteins, and those proteins then also are subject to, at least in theory, to regulation. All of which must be somehow correctly controlled, where there are mechanisms of control, to get you the final active protein. Lest I leave something important out, the proteins that are produced will also be degraded, and if you have ways of controlling the rate at which proteins are degraded, you have yet another way to control gene expression in the broadest sense of how much of a polypeptide is actually going to be present in a cell at any given moment. So in practice, it turns out where you have regulation, those genes that are regulated, most of them are actually regulated at the level of transcription, at a simple switch that turns genes on, turns them off, or in some cases it's like a dimmer switch. You can increase or decrease the level of transcription without actually shutting the gene on or off. But it's almost always related to how often transcription is initiated by RNA polymerases. So let's look at a couple of ways the genes in cells are actually regulated. Chemical signals that I've already told you about are things like hormones and growth factors. Another group called cytokines would include things like the interferons. You may have heard that in another context, viruses induce the production in our bodies of things called interferons, which were called that because they seem to interfere with viral growth, viral replication. Well, interferons are cytokines. Interleukins are another kind of cytokine. You'll learn more about these things if you take courses in neuroscience or in physiology. Here I just want to mention that there are some of these that our own bodies produce for the purpose of making other cells respond. And very often the response, not always, but very often the response is to control gene expression. Now, chemical signals outside of a cell, can convey information to the cells either directly, that is to say by getting into the cell, interacting with a receptor molecule, usually a receptor protein in the cell, and then going about the business of getting a cell to respond, or by signal transduction. And this is the case for many cytokines, growth factors, and hormones that are too big or too highly charged to actually get inside the cell. And so you may recall that these kinds of molecules are likely to have cell surface receptors to which they bind, and that binding elicits a response inside the cell, accomplished by this process of signal transduction, which, by the way, if you remember, will always involve changes in the conformation of proteins, in this case the conformation of membrane proteins. And those changes in shape of membrane proteins elicit new enzymatic and other binding activities that eventually convey a signal from outside the cell to inside the cell. So let's look at the direct action of some hormones, such as steroid hormones. Here we have a steroid hormone. Because it's a lipid, it gets inside the cell. Let's look at what happens again. Here it goes. It gets in the cell. It binds to a protein, a receptor, and we know that that receptor dissociates from another protein component. So it's actually a protein with two parts. One part, when it comes off, frees up the other part, now still attached to the steroid hormone, to cross the nuclear envelope, get into the cell, and bind to double helical DNA at DNA sequences specifically recognized by the hormone receptor complex. So the steroid hormone receptor sits there. Let's look at it one more time. It sits there just in waiting, either in the cytoplasm or sometimes they're actually in the nucleus. This one is in the cytoplasm, gets into the nucleus, bound to the hormone, because its conformation is such that it can be transported into the nucleus and bind to DNA. And if you're going to turn genes on, you better be able to bind to the DNA somewhere near the gene. And so we're assuming that this is what's happened here. That induces, in this case, the transcription of a message, which of course gets out into the cytoplasm, associates with ribosomes, which then translate the message into a protein that was not being made before. Steroid hormone receptors are, in fact, transcription factors. Let's take a look at an overview of signal transduction. And this is, again, by hormones or substances that cannot get inside cells. And we recognize two fundamentally different mechanisms by which such signals or hormones would work. So let's look at one of them right here. This is one. The hormone binds to a membrane receptor, changes its shape, which allows it to interact with another protein in the membrane. Let's look at it again. The hormone binds to the receptor, which changes shape. That results in an affinity for a membrane protein, which is an adenylate cyclase, which is an enzyme that catalyzes the conversion of ATP to cyclic AMP. Cyclic AMP is the classic second messenger. It's also the first second messenger that was found. So we've just seen signal transduction. The hormone, the red pentagonal shape, is still on the outside of the cell. It's bound to a receptor on the surface of the cell. But its signal, its information, has been transduced cyclic AMP, a second messenger, which then bound to another molecule, in this case another protein, characterized here by the black diamond. That protein is a protein kinase, which means it's going to catalyze the phosphorylation of some other protein. Let's take a look again. Here we have cyclic AMP bound to this protein kinase. ATP is the source of the phosphate to phosphorylate another protein. Now, I'm not showing you the details here, but each arrow represents a phosphorylation reaction in which another protein is being phosphorylated by the previous one. So we talk about a kinase cascade or a phosphorylation cascade initiated by the hormone in this example. The hormone could be glucagon or adrenaline, which does pretty much exactly what you're seeing here with some more details, but the same thing. So each of those red arrows is a cascade of phosphorylations in which enzymes can get phosphorylated one after another, and the last one in this cascade has the ultimate cytoplasmic effect. Again, as an example, if this were adrenaline and this were a liver cell, the result of the several phosphorylations is to activate the enzyme glycogen phosphorylase. So that's the enzyme that's going to catalyze the degradation of glycogen. If you think of adrenaline as the fight or flight hormone, the reason it would do this is you've just had an adrenaline rush for whatever reason. The adrenaline has gone through your circulation to your liver, and it's going to let your liver release a whole lot of sugar so you can play better tennis, escape the robber, whatever it is that you need to do that caused the adrenaline rush. Here we have gene regulation. The other pathway for this enzyme is to create a phosphorylation cascade that, again, phosphorylates one protein after another. In this example, we end with the phosphorylation of a protein called STAT. It's a signal transduction and activation of transcription protein. That's what STAT stands for. And that phosphorylated protein, which we just saw, let's try to see if I can do it again. There's the STAT protein without a phosphate. The result of the phosphorylation cascade is to phosphorylate the STAT protein in the nucleus, which now again has affinity for DNA in the vicinity of a gene that needs to be turned on in response to whatever that hormone was. And we see that happening by the induction of transcription, the activation of transcription. The transcript then again leaves the nucleus, goes into the cytoplasm. There, of course, it is transcribed. Well, this is one kind of signal transduction. Let's look briefly at the other. The hormone is a polypeptide. Many growth factors act like this. They bind to a receptor on the surface of the membrane, which is also a kinase. So it's a receptor kinase. And the result is ATP donates a phosphate and actually phosphorylates the receptor itself on the cytoplasmic surface. And that then kicks off another phosphorylation cascade, and once again, the result is to phosphorylate a nuclear protein, which is a transcription factor, which can bind to DNA and turn genes on. So cell membrane receptors transduce an external signal into some small molecule in the cell, like cyclic AMP. General levels of phosphate can be elevated, certain lipids can be elevated, or calcium. All of these are other second messengers, and they all facilitate a phosphorylation cascade that leads ultimately to a final phosphorylation and a final cellular response in the cytoplasm, as we just saw, or in the nucleus. And again, we're focusing on gene regulation, so we're really looking at signal transduction events that lead to gene regulation. So, we talked earlier about proximal and distal DNA binding sites for transcription factors. So let's look at DNA and see how these are organized. Now remember that most eukaryotic genes are split genes. They have exons and introns. Here's a piece of DNA alongside a gene. We see the start site of transcription. We see the promoter. This is the sequence, usually it's called a Tata box, along with associated elements that form the promoter. And then just upstream, and this could be 20, 30, 40, 50 nucleotide pairs upstream, we say to the left of the gene, would be a promoter proximal DNA sequence, a promoter proximal regulatory element, meaning this is capable of binding some transcription factor that's going to facilitate turning this gene on. Let's zoom out a little bit and look at the same piece of DNA, but this time let's look at promoter distal elements as well. You can see that there are regions, so-called enhancers, which are just DNA sequences that can bind to transcription proteins, transcription regulatory proteins. They were first identified as distal elements and called enhancers because when they were activated by their transcription factor, levels of the transcripts of specific proteins was elevated, and so, or enhanced, and so that was one of the effects. Introns, these often very large regions of DNA between the coding regions of the gene, are also prime places for enhancers. So here we have a promoter proximal element showing near the promoter and a handful of enhancers or promoter distal elements. And they can all interact, and many genes, by the way, have more than a few gene regulatory DNA sequences, and they therefore interact with more than a few transcription factors. To get RNA polymerase itself to bind to the promoter and form the transcription complex, that requires several different proteins by itself. These are sometimes referred to as promoter proximal regulatory proteins. Transcription factors that bind to enhancers, look what they do. They actually result in bending the DNA, or bending chromosomes actually, because at this point they're not naked DNA, they may be loose, euchromatic DNA, but they're still associated with lots of chromatin proteins. So what happens is you bend the chromosomal DNA, or the chromatin DNA. Remember that the cap protein in bacteria bent what is essentially naked DNA, and in so doing, it loosened up some of the H bonds between the two strands, making it easier for RNA polymerase to get in and bind to the strand that it would actually have to transcribe. If you could bend eukaryotic chromatin or eukaryotic chromosomal DNA, you might achieve the same purpose. And that is one of the functions of some of these promoter distal enhancers and their transcription factors. So here's a picture of chromosomal DNA. It's not showing the chromatin as such, but it is showing the DNA bent over in the vicinity of a gene and complexed with a whole host of proteins, some of which are going to be intimately involved in the actual act of transcription, and others which are involved in regulating the gene, making sure that it is on, and in doing so, actually bending the DNA and making it more accessible to the polymerase and a variety of other proteins in the transcription complex. So let's take a look. There are a couple of enhancers. They're actually shown as being on opposite sides of an entire gene, which is not unusual, and they are bound to some proteins shown here as well. And the effect of binding their transcription factor proteins is to bring those two enhancer regions together, in other words, to bend the DNA, and to interact as well with components of the transcription complex that assembles at the promoter. So let's see what other proteins we have here. So for example, this could be a steroid hormone receptor complex, or it could be a phosphorylated transcription factor such as the activated STAT protein I spoke about a moment ago. There's our promoter. There's a protein that helps the RNA polymerase situate itself in the right position so that transcription can begin. This protein is called the TBP, or TATA binding protein. Remember that the promoter has a sequence in it, in most cases, of TAA, or the TATA box. And so the TATA binding protein is one that is designed to recognize and bind to that sequence, and so in so doing, allows other components of the transcription complex to assemble. Eventually, the last of these is in fact the RNA polymerase. So there's the complete transcription complex. Once the RNA polymerase is on and has been modified somewhat, it can begin transcription. All of this would not happen were it not for the presence of enhancer proteins. In other words, this gene would only be active if those transcription factors that bind to the distal elements were actually there and doing this. Oh, there's our RNA polymerase 2, the RNA polymerase that in eukaryotes transcribes messenger RNAs. Just a word about the shapes of proteins. I make a big deal in my classes about the shapes of things. There are several proteins here that interact with DNA, and there are several shape motifs that are common in proteins that interact with DNA. One of them is the so-called zinc finger protein. This is a protein that is associated with zinc ions, peppered as it were throughout the protein. And then there are these helical regions that project from the protein and get into the major groove, as you can see here, the major groove of the DNA. That's the business end of the zinc finger molecule when it binds to a sequence it recognizes. Another example of a DNA binding protein is the helix-turn helix motif. This is characteristic of a number of bacterial gene regulatory proteins. And you can see the helix, the turn, and then the other helix, and you can see the helices that project from this and actually also penetrate the major groove. So those penetrating helices at the bottom are what are really recognizing specific base sequences in the DNA. And the last that I want to show you is what's called the leucine zipper protein. The leucine zipper itself is a way of holding two separate proteins in a dimer together. Leucine is a hydrophobic amino acid, and if you have a regularly spaced bunch of leucines on two strands, they will tend to come together and zip up like a zipper. And the part of the protein that is extending from either half of the zipper again, are helices that penetrate the major groove and are responsible for binding specific DNA sequences. So that brings me to the end of an overview of eukaryotic gene regulation